Hello, my name is Professor Eric Brenner, and I am your instructor of record for the Principles of Biology Laboratory, and I'd like to introduce the first lab to you. The title of the first lab is The Art of Pipetting, and the reason why we have a lab devoted completely to pipetting is because it's very important. In today's molecular world, many of the manipulations are done on a very small scale, and so we have specialized equipment to do that. And these are the pipettes. Why are we so worried about pipetting? Well, because if you do not pipette correctly, your experiment will not work. And so, just like many things, you want to develop good habits from the outset so that your experiments go very well. And because your lab period is so limited and resources can be very expensive, if you have good pipetting habits, then we won't waste materials and your time will be used efficiently. So let me introduce to you the pipette. These are called micropipettes, and we have three sizes. And depending on which lab, you may have all three or a subset of that group. So this is the P1000, okay? And it's called P1000 because it takes 1,000 microliters, and 1,000 microliters is equal to 1 milliliter, okay? And so the way to use a pipette is, first of all, get a feel for how it is in your hand. So here's a pipette. You rest it in your palm with this little hook around your finger, and it has a plunger on the top, okay? Now, it turns out that there are 2 stops on every pipette, okay? This is very important, and the 2 stops have different amounts of really energy you need to put in to reach that. So when you go down to the first stop, you push the plunger in, you'll feel some kind of resistance. You've reached the first stop. Let's bring that plunger back up. Okay, when I go back and forth to the first stop, that's the actual volume that you will be pipetting. Okay? Now the second stop, go down to the first stop. Okay, so that's the volume you've just pipetted. When you push through to the second stop, that is the residual that gets caught on your tip. And I'm going to explain what that means to you in a moment. Okay, so let's talk a little bit more about the volume. Okay, each pipette here has a little screen which demonstrates how many microliters you're pipetting. Now the P1000 is accurate within the range of 100 to 1,000 microliters. Okay, that's 100 to 1,000 microliters. So let's say I wanted to pipette 22 microliters, I would not use this pipette. But if I wanted to pipette 400 microliters, I would use this pipette because that fits within the range. Okay, and the way to adjust the volume is to simply turn it. Okay, and as I turn it, I'm going down, and I don't know if you can see this or not, I doubt it. But what matters is that when you actually look at the little window here that shows you the volume, that when I turn it, I can actually reach 400 microliters. So let's say I want to pipette 400 microliters. I turn until I reach that specific amount, 400 microliters. Okay? So I hold it as I told you, and now I need to get a pipette tip. So we have here special boxes which allow you to do that. You open it up, and we have our pipette tips. Very self-explanatory. You push the pipetter in. It catches a tip on it. It's a nice solid hold. I can pull it off if I want. Push it back on. Okay. Now if I'm going to pipette that volume, 400 microliters, then I go over to whatever source is going to give me that volume. So here we have a microfuge tube. Okay. And what you do is you just pop the cap, and you may be wearing gloves depending on what you're working with. In this case, I'm working with water, but let's say it was some kind of chemical, you'd want to wear some gloves. And you pop the top, and you just very carefully open it. I've taught myself to do all this with one hand. And then there's a little volume of liquid. Before you go into the liquid, you want to push down to the first stop, because that's the volume I'm pipetting. Okay? As I put the tip in, I just want to slip under the meniscus and slowly collect the volume. And notice I even move the tip down as we go. Voila, 400 microliters. Okay? I want to do that again. Just make sure we all understand. Okay? Okay, let's start from the very beginning. I push down to the first stop. I go over to my volume. I collect that liquid. And I have 400 microliters. Okay? Now I want to pipette this. I want to transfer it over to another tube. So let's say I grab another tube. I'm going to use this tube. I bring it over and I touch the tip to the end because I need that kind of adhesive forces to help pull that residual out. And as I pipette, you'll see it going out and I've reached the first stop. But guess what? There's a little bit left. That's what the second stop is for, because now as I push it through that greater resistance to the second stop, that last little bit comes out. And that's necessary for accurate pipetting. Now don't leave the tip in when you release the plunger. No, 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 no. Keep past that second stop, take it out, then you can release it. Why? Because you would suck out some of the volume. You wouldn't want to do that. Close the cap, and you're done pipetting. Go to the garbage, eject your tip, right? And you're done pipetting. Now, we have P200, which is good from 20 to 200 microliters. So let's say I want to pipette 100 microliters. Okay, I'm going to rest in my palm just like I did before. But I'm going to use a different set of tips. And these tips are good for the P200 and the P20. I'm going to open it up. Okay. I'm going to get myself some tips. A few have spilled out here, as you can see. Catch it just like I did before. And all of this, the first time you do it, you want to do it very slowly. Okay. And I'm just going to pipette as I did before. And you can learn to pipette.